And so from a mechanistic standpoint, I just kind of wanted to bring this, bring these concepts forward. I'm not sure if in other modules you would get this pathophys, and so this is my background, so I'm gonna do it my way. Um, some of the mechanisms that innate immune cells can promote cancer seem somewhat foreign and probably contradictory based on what you've already learned. And so here's a few examples. And so here's two direct mechanisms whereby normal, or skewed innate immunity can actually promote cancer. Okay, so here you can see induction of DNA damage by the production of free radicals. The process of a persistent immune reaction can generate free radicals in high concentrations or in concentrations that are not adequately maintained by the cell physiology. You can actually promote DNA damage. There's a paracrine regulatory pathway that's quite potent and exists through the NF-kappa B pathway, which is a very long, complicated signal transduction pathway. But those two pathways have been directly capable to kind of promote innate immunity to actually promote cancer phenotype. There's a few indirect mechanisms. The promotion of angiogenesis can actually be produced through several cytokines and chemokines. That's my research background. We actually were able to show how macrophages, when exposed to a quite potent chemokine gradient, actually became re-educated and actually allowed tumor cells to metastasize to the sites of the macrophage residence, which was really kind of weird. COX-2 has been shown to be upregulated in tumor cells, which is why there's this push to give patients aspirin. And there's been a kind of a global suppression of an anti-tumor immune response. And so it's been well documented that patients that have developing tumors produce antibodies against the tumors as a course of normal immunity. But it actually seems that the antibodies that they produce are defective and of the wrong type and may actually promote tumorigenesis because of the way that they bind to some of the tumor antigens and then the way they actually signal. And so if you're at all interested in any of this, there's a really great paper which is cited here at the bottom, Paradoxical Roles of the Immune System During Cancer Development, published in Nature back in 2006. This is a great paper. Obviously it's a little dated, but it's been referenced, God, over I think maybe 1,500 to 1,800, just shy of 2,000 times. It's a really good paper. So anyway, so enough about that pathophys. Let's get into some of the more details. And so other mechanisms by which the adaptive immune cells can promote cancer are listed here. You can see alterations in anti-tumor cytotoxic T activity. They actually become redirected, and so they ignore the tumor cell. Indirect mechanisms, Tregs have been shown to play a huge role. Remember, Tregs are the suppressor cells, and so even though you may get a potent cytotoxic T lymphocyte infiltration, those Tregs will actually turn that process off and literally allow tumors to grow unchecked. And so, of course, there's research going into kind of switching some of these outcomes and reprogramming some of these cells, but nothing's been done specifically. Okay, so that's kind of a brief overview of the physiology, of the pathophysiology, at least of what we know. Let's look at some of the ways in which we can class these drugs. Depending on who you read, will give you different pharmacologic classes, and I'm going to give you both. I particularly don't care which classification system you use. I really just want you to know how they work. This is a little bit different than some of the other drugs, which you have to know the class as well as the main players. But this is a very complicated literature and a very specialized literature. So I'm more interested if you know what the drug does and how it does and what types of diseases it can be used, rather than which classification scheme it is. This one really isn't too far off because we're actually classifying them on the mechanism of action. The other drug classification schemes are discussed in the next slide. So we can talk about drugs that inhibit gene expression. Of course, those would be steroids. We can talk about drugs that can change the lymphocyte population via depletion or expansion. We can look at signaling pathways that are typically blocked in order to curb that expansion process. We can use neutralization processes to turn off cytokines, especially the really potent cytokines that are driving this immune reaction, and some of the other drugs that are listed there. So that's the first classification scheme. How about another one? We can also look at sites of activity, and this is how I've done this particular lecture, because I feel like, with these mechanistic standpoints right here, there is actually some overlap depending on which thing you're blocking and how it works. So there's a few drugs that would actually be in several of these classes. Based on the sites of activity, makes a little bit more sense, at least more conceptually, because you can see a B cell target, you can see a T cell target, and so on. So this is the classification scheme that I used, and it's a little bit different than what we've traditionally done in pharmacology, but these are not traditional pharmacologic agents. Most of these drugs are proteins, actually. Okay, and actually along that line, most of them are antibodies. Okay, and so this is just kind of a quick review. Why would you use an antibody as a drug? What mechanism does a normal functioning antibody have that would render it even remotely possible to be a drug? Okay, and here's a good example. This is a CD20 molecule, which is a good marker for B cells. We don't really know what CD20 does specifically, but we know if a cell has CD20 on it, it's a B cell. Okay, so we've produced an anti-CD20 molecule, and what it does is it actually kills the cells that it binds to, and it does so through at least 3 different mechanisms. And the first one here shown on the right is this antibody-dependent cell-mediated cytotoxicity mechanism, okay, which uses basically the adaptive, the innate immune cells, the macrophages, and the natural killer cells to basically put a little flag on the target cell and attract some of these more potent killing cells to this B cell. And so you can see that process here. I hope you can see my mouse, where this green cell was attracted because this antibody bound to this cell, and then this green cell killed this B cell. Okay, and this B cell is this reddish cell that changes to pink as it dies. Complement cascade basically has a similar outcome, where the complement binding activity is essentially the killing phase, where the membrane attack complex is initiated, and then that kills that renegade B cell. Okay, and then of course the third method is direct apoptosis, where this antibody gets basically internalized and degraded. The B cell signaling pathway is shut down, and the B cell undergoes apoptosis. So those are the 3 different mechanisms where a monoclonal antibody, which is this anti-CD20 molecule, this blue monoclonal antibody, can actually shut down a cell process to promote apoptosis. Okay, and so this is just a nice summary slide. This is showing the naked antibody itself, so just the antibody, can produce antibody-dependent and cell-mediated cytotoxicity, as well as the complement-dependent cascade, and that will directly kill the tumor cell or whatever cell is being targeted. It doesn't matter. And then you can also decorate that antibody, that monoclonal antibody, with biotin. And you can have this biotin-streptavidin reaction. You can put a marker sequence on it to attract killer cells. You can put a liposome on it so that this thing gets internalized and degraded. You can even put a drug that can be activated upon binding, an immunotoxin, an immunocytokine, and so forth, radioligand. And these types are called immunoconjugates, where you put some other type of decoration on the molecule, and then that renders this process even more toxic. Okay, so all of these monoclonal antibodies will, in fact, somehow kill this. And this could be a tumor cell, this could be a renegade B cell, this could be a renegade T cell. It doesn't matter. If the marker's here and the antibody binds to it, it will kill this target cell. Monoclonal antibodies have a weird and complicated nomenclature. Everybody says, when they first start studying this stuff, who the hell names these drugs, and why do they name them cetuximab or rituximab and so forth? Well, there actually is a method to the madness, and that nomenclature is described here. Basically, it's based on the International Non-Proprietary Nomenclature, the INN. In general, most of these monoclonal antibodies had the suffix or the stem at the end, MAB, monoclonal antibody. And then there's substems, and so the part that's in front of it, if it's ZU, that stands for humanized. So the primary scaffolding is a human molecule, but then there might be some mouse or other components to it. If it's OMAB, it came directly from a mouse. If it's AMAB, it came from a rat. If it's EMAB, it came from a hamster. IMAB, it came from a monkey, and so on and so forth. And then the other substems were supposed to be based on the disease or the target class. That's becoming less useful, as is the other substem that's supposed to denote which type of tumor it originally targeted. The reason that became less useful is as the research focus broadened, we found out that we can use some of these monoclonal antibodies for other diseases besides the original intent. So a good example would be rituximab. Rituximab is the anti-CD20 molecule, and it binds to specifically B cells and eliminates them from the circulation and also from having any activity. And now we're looking at rituximab for solid organs. Solid organ tumors. A great example would be lung cancer. There was a clinical trial looking at rituximab for lung cancer. And so there isn't really classically a CD20 component to lung cancer specifically, but there is a B cell component to lung cancer, and that's where some of these drugs have crossover. Okay. And so in terms of looking at the structure of some of these molecules, or some of these monoclonal antibodies, this is really the difference. So, here's the human one. Obviously, it's completely human. Humanized means it's mostly human, but there's some mouse hybridoma, especially in the VGF region. Chimeric antibodies have a little bit more mouse structure in terms of their binding sites. And then, of course, the mouse monoclonal antibody is completely mouse and not human at all. Okay, and so depending on how much mouse or how much human constitutes the antibody governs the name of it. Okay, so that's kind of a brief rundown of monoclonal antibodies and some of these disease-targeted approaches. And so now let's get into the different drugs.